Okay, thank you very much. I guess I'll be the last to speak in this conference, and I hope I don't ruin it with my talk. I'm just going to describe some work that has been recently going on in my lab and in collaboration with a number of others, namely Ed Susko, Laura M., and Dan Gaston. And it came about mainly because of this big outstanding problem we have in understanding, the origin of eukaryotes. For those of you who aren't totally familiar with the idea of eukaryogenesis, it's the origin of nucleated cells from prokaryotic precursors. This probably happened sometime between 1.6 and 2.5 billion years ago. And it's a big, difficult problem because there's very little in between grades of cells known between prokaryotes. So shown here is the sort of latest idea of what we know about eukaryogenesis, that the closest sister group to the host lineage that gave rise to eukaryotes are the Asgard archaea. And shown here in this tree is the first, what people call the first eukaryotic common ancestor, the beginning of the branch leading to eukaryotes, and then the last eukaryotic common ancestor. So that's the common ancestor of all crown eukaryotes. In between there, a lot of things happen: gene duplications, the origin of the nucleus, meiosis, mitosis, flagella, all sorts of things. And there are really no known intermediates between that. And that's what's shown here is a bunch of extinct lineages. The fossil record is also very difficult to interpret. The most important thing from this talk is really the point of when the mitochondrial symbiont arose by endosymbiosis in that host lineage. So we know that mitochondria from all eukaryotes derives from a free-living bacterium. We know it's an alpha proteobacterium, or closely related to alpha proteobacteria at least, but we don't know whether that symbiosis took place very early in eukaryogenesis, in the middle, when some of these other features had evolved, or very late in eukaryogenesis. And ordering the relative timing of mitochondria versus these other innovations has been really difficult. In 2016, Pittis and Gabaldon had an interesting idea that maybe they could look at genes of different origins in eukaryotes, different prokaryotic origins in eukaryotes, and get an idea of the relative order in which those lineages evolved, those sort of genes evolved by looking at their so-called stem lengths. So if you think of each of the genes coming from different kind of ancestry, you have the theca, the host lineage genes that share a common ancestry with Asgard archaea. And then there's mitochondrial genes, which are genes that came in with the mitochondrion. And if that event took place, the mitochondrial origin took place much, much later, you might expect that the stem lineage between the closest sister group of the donor lineage and the last eukaryotic common ancestor might be shorter than if it were coming in very early on. In that case, it might be more comparable to the age of the host origin genes. And so you can see here this stem lineage is depicted here as the lineage between this common ancestral node with the closest sister group and the last eukaryotic common ancestor. So Pittis and Gabaldon collected 1,000 protein families in eukaryotes that they could trace to the last eukaryotic common ancestor, determine their closest sister group, and then tried to look at the distribution of these stem lengths to see if there were consistent patterns with regard to those. So the general idea here is, as I was saying, that if the alpha proteobacterium came in late, you might expect that the branch lengths leading to this last eukaryotic ancestor would be very short relative to genes that came in with the host lineage. Alternatively, if the mitochondrion came in very early, you might see a more comparable branch lengths with these mitochondrial genes versus the host genes. So that's the motivating idea behind it. So they collected about 1,078 of these proteins, determined the prokaryotic sister group for each phylogeny of each of these proteins, and then basically tried to motivate the idea of using the branch lengths in the following way, that branch lengths are related to the time span of branches through some overall rate of evolution and the time span of the branch. And so they thought perhaps if different proteins had different rates of evolution intrinsically over the whole tree, you could normalize and remove the effect of that overall protein rate by taking a ratio of the branch length of the stem to branch length within the eukaryotic line of descent. So they defined normalized stem length here as SLG as the branch length of the stem divided by, in this case, they took the median from root to tip distances on each protein tree within the eukaryotic subtree. So here, little s denotes the stem, and little e denotes the eukaryotic part of the tree. Okay, so the idea here is that this will be equal to something along these lines: the rate times the time span over the median rate times the time span of eukaryotes. And if this Rg is roughly constant, then this stem length will reflect the ratio of the stem branch length to the eukaryotic branch length. So if you now plot distributions for these stem lengths for genes of different origins, systematically smaller values of the SLG mean that the time span that they spent in that eukaryogenesis phase was smaller. So TS was smaller for those genes. So that's the general idea behind it. Just to reiterate with another diagram, this is the actual time tree on top, and this is the time span that we're after, the time span that the gene G that was introduced into that lineage spent during eukaryogenesis. This is the eukaryotic time span, and what we're getting from each gene tree is actually the branch lengths, this LS, G, and this median of the root to tip distances, branch lengths of the eukaryote subtree. So that's another way of looking at it. So they did this, they collected those proteins and plotted, they did a number of different things. I'm going to focus here on just one main result that they presented. And that is a box plot of these normalized stem lengths, broken out according to the sister group lineage for the eukaryotes. So here we have the archaea, so this is the host lineage. Bacteria, these are bacterial genes that are not of alpha proteobacterial origin, so not mitochondria, but of other origins. And then these presumably are the mitochondrial origin proteins. And what they found was that the stem length distribution was shifted to be much larger for archaea versus bacteria and alpha proteobacteria. And so they concluded from this that mitochondria were relatively late in eukaryogenesis. So the stem lengths here of mitochondrial genes were quite a bit smaller than the stem lengths on average of the archaeal genes. They also concluded that there was other bacterial taxa that have given rise to core eukaryotic systems, but we won't discuss that in detail in this presentation. Okay, so that's all well and good, except it wasn't very long before a very stringent criticism popped up by Bill Martin's group, published here in Genome Biology and Evolution, where they claimed that the claims of what Pittas and Gabaldon had said were actually false. That's a pretty bald title there. Late mitochondrial origin is an artifact. And their claim was that you really can only justify, well, they made a number of claims in this paper, but with respect to the main point of my talk, their claim was that Pittis and Gabaldon's analyses are only valid if you have a strict molecular clock. That is, you have to assume that the rate of evolution on the stem branch is equal to the rate of evolution on the eukaryotic branch. Otherwise, you won't get those stem length ratios reflecting times of origin. The other option is that they are a fixed ratio, that somehow there's a specific ratio of stem length branches versus the rates in eukaryotes. But either way, this is a strong assumption. And then when they looked at the actual trees in the Pittis and Gabaldon data set, they found that many of them were very, very inconsistent with a strict molecular clock. And you wouldn't really expect a strict molecular clock operating over billions of year timescale. So that was the criticism. There was some back and forth about this. This is something that came to our attention a while ago, and then we recently revisited it in collaboration. And this is work mainly done by Ed Susko, but in collaboration with Mike Steele, where we really asked the question, is that true? Do you need a molecular clock to justify this Pittis and Gabaldon approach? And it turns out, kind of surprisingly, that you don't. What you do need is an assumption that the rate of evolution along branches for protein genes, for any protein gene, is a continuous time stochastic process with an overall protein rate, Rg, and a time-dependent relative rate, little rg, which is dependent on time on branch S. This continuous time stochastic process can be doesn't have many constraints on what it needs to actually be. It just has to be bounded away from 0 and infinity. And all genes at a given time point have to be independent draws from that rate process. Anyway, in the paper that we've submitted to bioRxiv here, and I'll put a link on the chat for that as well at the end. Ed showed that the probability that you will get a branch length from group A genes versus group B genes of different origins will be equal to exactly 1/2 when the time of origin of group A and group B are exactly equal. Okay, so the probability, this sort of makes sense. If A and B genes are both getting drawn from the same stochastic process, these are just random variables coming from the same stochastic process with the same marginal distribution. So you expect that any random choice of one versus the other has a probability of 50% of being greater than the other. Okay, and the probability that this is greater than that is greater than 1/2 only if the time of origin of A is greater than B. So this way of phrasing it is important because this is exactly the form of the statistical test that Pittis and Gabaldon were using, the Mann-Whitney U test. This is the null hypothesis for testing two distributions of A and B under the Mann-Whitney U test. This is the alternative hypothesis showing that one SL distribution is shifted relative to the other SL distribution. And again, that's described in great detail in this bioRxiv paper. So that comes with some assumptions, though. The assumption is, as I said before, that this rate process is somehow bounded away from 0 and infinity. That's not too surprising if you think about mutation rates can't exactly be 0 or infinite. Proteins within groups A and B have the same times of origin within the groups. That is, all of the genes in group A should have the same time of origin. All the group in group B should have the same time of origin. That actually can be relaxed, and that's actually a complicated little point that's discussed in the paper. But the one that I want to focus on is this assumption that all proteins are independently evolving according to the same rate process, RG. And that is the problem that I think actually starts to manifest in this particular analysis. So I'm just going to give you an example of how such problems could exist. And I'm just going to show you a piece of an alignment from the elongation factor 1 alpha protein. This protein here in blue is a core part of the translational apparatus of all cells, bacteria, archaea, and eukaryotes. And it delivers the amino-isolated tRNA to the ribosome during protein synthesis. So if we zoom in on a piece of the alignment of the archaeal versus eukaryotic protein here, you'll see some interesting points. Now, just keep in mind that the time span for all of the eukaryote sequences in here is probably over 1 billion years, and same for the archaeal sequences. So there's been a lot of time passed within each of these groups. And you can see quite different patterns of evolution in the two groups. So, for example, some sites are going fast in archaea and slow in eukaryotes. In fact, there's quite a lot of differences. Some sites have very different physical properties. For example, this one has valine isoleucine. This one just has threonine. Those are quite different. This is hydroxylated. These are hydrophobic. Okay, so this is an example of what we and others have called functional divergence. That is that the rules by which the proteins are evolving have actually shifted between archaea and eukaryotes. And this isn't too surprising because the eukaryotic EF1 alpha actually has additional functions that are not found in the archaeal EF1 alpha. So at some point in the past, during eukaryogenesis, some of those new functions were acquired and changes in the dynamics of the evolution sites occurred. So what if a functional divergence has differentially affected some proteins in the Peterson-Gabaldon data set versus others? And this is something we investigated. This is work done by student Dandan Zhao and Katya Radan in collaboration with these 3 fine individuals. And so what we did was we have a piece of software that we call Fundy that investigates functional divergence. And we analyzed a large proportion of the protein families in Peterson-Gabaldon for functional divergence. The reason this is not all of them is because in the rest of the proteins, there were less than 3 taxa in one of the 2 subtrees, and our tool can't use any fewer than 4 taxa in both subtrees. So we used our Fundy tool, which is based on a phylogenetic mixture model for the identification of functionally divergent sites that we published in 2010. Sorry. So basically, Fundy is a mixture model. It models sites as being either functionally divergent over a given split in the phylogenetic tree or non-functionally divergent. And it has a proportion associated with that with each class. And that is this rho parameter. And that tells you to what extent functional divergence is affecting sites in a protein across a given phylogenetic split. This is implemented on top of standard site heterogeneous profile mixture models, the LGC20 plus F plus gamma model, and we can estimate the optimal branch length for the split of interest. In this case, it's going to be the stem split between eukaryotes and their prokaryotic relatives. So we can optimize that in the presence of functional divergence. So just to give you a brief background in this site heterogeneity process, the LGC20 type models, normally people have widely used these site homogeneous models that assume that all sites from an alignment are coming from a single substitution model that is given by the product of stationary frequencies and an exchangeability matrix. So the model, substitution model Qij going from I to J, instantaneous rate, is say in this LG plus F model, just a single model for all sites. In site profile mixture models, we assume that sites come from any number of different classes. And those different classes are characterized by different stationary frequencies of amino acids. So here we have 4 different profiles, that is frequency profiles of amino acids. And we assume that those stationary frequencies vary across these classes. We assume they all have the same exchangeabilities, and so that we now have as many Q matrices as we do classes. And the way this is handled in a mixture model context is that we take the site likelihood, the probability of the site xi, conditional on a given site class. Here we have pi c indicating the site class, and a rate class coming from the gamma model. And we sum up that site likelihood over all possible site classes and all possible rates coming from the gamma distribution. So it's the weighted sum of site likelihoods coming from each of these different classes. So we don't know which site comes from which class, so we do a weighted mixture of them. And that gives us our site likelihood. So Fundy is on top of this. So it takes that site likelihood and models it as 2 things: a functionally divergent set of sites and a functionally non-divergent set. Okay, so this here is the normal way in which a likelihood would be calculated over the entire tree. For the functional divergence part, we assume that basically the sites are unlinked now with respect to their history. They're almost like different, the 2 subtrees come from different alignment positions. And so the likelihood is now just the product of the likelihoods on each of the subtrees. And this turns out, even though this is not a mechanistic model for functional divergence, it turns out to capture functional divergence very well under simulation. So this part of the mixture is the product of the likelihoods of the 2 subtrees, and this is the regular likelihood over the entire tree. And then this row parameter controls what proportion of sites, or the probability that a site is coming from one class versus the other. Okay, so going back to the Peterson-Gabaldon data set, this is just a reanalysis of their data set using the LGC20 plus F plus gamma model. And again, we find, just like they did, that the archaeal stem lengths are much larger than the bacterial or alpha proteobacterial, and in Mann-Whitney U-tests, they're significantly greater. However, if we start estimating for those groups of proteins, the row parameter with Fundy, using the Fundy mixture model, we find that in fact, the archaeal proteins are enriched in proteins with more functional divergence than the bacterial proteins or the alpha proteobacterial proteins. So they actually tend to be more functionally divergent. And if we look at, say, the correlation between the logit transform of the row here, the proportion of functionally divergent sites versus the stem length without Fundy, for example, previous to using this functional divergence model, we get actually a very strong signal that the stem length is strongly influenced by sites which are undergoing functional divergence. So longer edge lengths tend to be more functionally divergent. However, if we now run the Fundy model to optimize that stem length, we actually remove the effect of the correlation between the functional divergence parameter and the normalized stem length. So Fundy has basically accounted for some of the extra branch length that is going on, that is over here, that is trying to accommodate functional shifts in the model. Okay, so now this is just box plots showing pre and post Fundy analysis of the stem length distributions. And you can see once you put Fundy into the analysis, the stem lengths shrink in all classes, and they actually tend to shrink a lot more for archaeal proteins than they do for the bacteria or alpha protobacterial ones. So accounting for functional divergence actually makes the stem length distributions of archaea bacteria and alpha protobacteria much more similar. However, there's still a slight increase in the overall stem length, or shift of stem lengths in archaea versus alpha protobacteria, even after you take into account functional divergence. So this, what this means on a functional level is that a lot of the proteins of archaeal origin in eukaryotes have actually undergone functional changes. This isn't necessarily overly surprising. Here is just shown parts of the large subunit of the ribosome, and it's well known that different ribosomal proteins, for example, have evolved along the eukaryote lineage versus the archaea, and that you can imagine that other ribosomal proteins that are in common between them would actually, now that they're interacting with other molecules, the dynamics of sites on the surface of those proteins is going to change because they have new functions associated with these other proteins. So there's a lot of functional divergence in the archaeal origin proteins, not as much in the mitochondrial origin proteins. So what does this mean? Well, this Peterson-Gabaldon method is actually a very interesting way of ordering evolutionary events. It's actually been recently expanded to looking at duplication events by Vosberg et al. In Nature, Ecology, and Evolution. So I suggest having a look at that. And Ed Susko's work has shown that it doesn't require molecular clock. It can be justified under a stochastically varying rate of evolution. The problem is that it assumes the process has to be independent over genes. And if some classes of genes are differentially affected by some process, such as functional divergence, that's going to ruin this justification for the Peterson-Gabaldon work. So we use this Fundy model to try and remove the effects of functional divergence to allow you to estimate the edge lengths without it. And that seems to help a little bit. We're left with the residual signal that archaeal edge lengths are longer than alpha protobacterial edge lengths. I'm not sure what to make of that. I think that's expected because archaeal origin ones must be older because the archaea was the host for the mitochondrion, but we don't know how much older, and we don't know how similar these edge lengths should be if they were contemporaneous. But the good news is that IQ tree is coming. We've got Min Bui on board to implement Fundy into IQ tree so that this kind of analysis can be done by other people. And again, we see this origin with Fundy, but this pattern Fundy of archaeal origin proteins still having a greater set of stem lengths, but it's really not clear what we should make of that. So I don't want to oversell it. So thanks very much to Dan and Katja, who did the Fundy analyses, Ed and Mike Steele, who did the mathematical heavy lifting with the bioarchive paper, and Laura M. and Dan Guston, who helped supervise and interpret the Fundy analyses with Dan and Katja. So with that, I guess I can take questions.